Okay, so my goal here is to give you the basics of the immune system, talk about some of the complications, but just to wrap up all the things. I know that activity I probably gave you last night or this weekend is a little bit challenging. And again, I gave you some lectures to look at as well, but just going to listen to me a little bit. All right, and we can write here. Okay, so we're starting this process. We talked about the innate immune system and the adaptive. Okay, and it should be clear the difference between the two. Okay, in fact, vertebrates are the only ones that have the adaptive. So the innate portion of our immune system dates back to single-cell organisms, okay, which are based upon the receptors on the outside of cell membranes. So essentially, our immune system, no matter if it's innate or adaptive, goes back to essentially identifying these receptors. Now, there's two types of receptors, and I'll get to that when I lay out the basics for the immune system that you have to know. Now, in the innate immune system, this is our first defense, and the first defense starts with a barrier, or a skin. If it's an opening, mucous membranes, okay? Antimicrobial proteins that attack the cell walls of certain microbes, or cell membranes of microbes, okay? Mucous membranes that will trap the sticky substance we have in our nose, okay, will trap certain. We talked about also other different kind of trap. We have cilia in our basically bronchial tubes here that push up mucus, okay, keep things moving. There's so many parts of this defense system in terms of the barriers. Now, keeping that simple, the barriers, we also have cells that are part of this innate immune system. And most of these are called leukocytes. Leukocytes. I'm not sure if I got those right, but leukocytes are a type of white blood cell. In fact, everything I'm going to talk about here in terms of cells are examples of white blood cells made by the mast cell initially in the red blood cell, the what do you call that? Hemipoietic, something like that, hemipoietic stem cell. And the name exactly, I lose the name there for a second, but it doesn't really matter. That stem cell makes all of these. Now, these leukocytes are a type of white blood cell that are part of the first defense, okay? And we have monocytes, okay? We have neutrophils. We have dendritic cells. Dendritic. There's eosinophils, there's basophils. There's a bunch of them. And these are the major players. Now, monocytes can move between cells and go through the bloodstream, but when they stick into connective tissue between cells, we call them macrophages. I know some people call them macrophages, but again, that sounds like a West Hampton name for me. So I'm going to call them macrophages, North Riverhead style. All right, so those are the same thing, they're just locked in a position, okay? Now, these are part of the first defense because these guys essentially detect a type of receptor on the cell membrane. Now, there's two major types of receptors. There's an MHC1, major histocomplex 1, and 2. Every cell that has a nucleus has that complex 1. That complex 1 is a unique set of, amino acids that stick from the cellular membrane that is essentially identical or unique for each organism. Okay? Now, person to person, that MHC1 complex is still very similar. Okay, although maybe a little bit different. And that's why when they match tissue organs or organ donors, they try to match people the best possible. It's not always perfect, but they can get a pretty close match. Now, on that MHC1, okay, if a cell is undergoing some problems, it's cancerous, producing proteins it normally doesn't make, because now it's undergoing some kind of, or it's under a viral attack where the virus has jumped into the cell and now is taking over the DNA to produce more of the virus. or a bacteria has attacked it, those cells start producing proteins that are different, and those proteins will get presented on the MHC1. And what these guys will do, for the most part, is, remember, this innate system is a broadband type of defense. They basically have receptors that basically look for that MHC1 to see if it's the same or if it's different. If it's different, as you've seen through other videos, it's telling you that that cell needs to die. That cell has problems. It's asking to be put to death. Okay? Whether it's a cancerous cell, whether it's a cell under viral attack, bacterial attack, or what have you, okay, it's going to put on that receptor something on top of it that's foreign that these guys can quickly, what? Identify. These guys also identify bacterium because they have receptors that are similar for a lot of the what? Bacteria and things that are out there normally. Okay? So these are the first basic defense. Yes?
But like, say if cancer cell somehow like doesn't change the outside, so like, or they like glimpse over like a sick like cell, is that how it gets like it spreads?
Well, a cancerous cell is going to start doing things it normally doesn't do. It's going to start churning out maybe some different proteins. It's going to start proliferating, growing, and doing things that the cell is not normal for its cell cycle. And that's going to basically put a flag on the MHC1 that these guys can quickly identify. That's essentially what it is. So when a cell's not doing its normal function, it can't help but take part of that new function and present it. All right? So that's where that is. But these guys are already in the bloodstream. They're already in between cells and an interstitial space. Okay? We call lymph. Okay? They're there. And if we have some kind of attack, for the most part, most of the time we don't even notice we're under attack because these guys will take care of it. But when they're overrun, and they're quickly overrun in some places, if you're under a viral attack, let's say you got some kind of cold or some kind of virus bug, you call it. Okay, these guys are quickly overrun because it's attacking what? A lot of the cells in all parts of your body. So these guys can be quickly overrun. So what happens here is, and it happens whether they're overrun or not, a couple of these cells, okay, these types, become professional presenting, antigen presenting cells. Now professional means on their MHC2, it's a different receptor. MHC1 basically measures how well that cell is. If there's no protein on it, it's a cell that's not going to be marked for death. It's left alone. It's got an MHC2 place. This is another set of proteins marker. Now what it does is, let's say a macrophage, or let's say a dendritic cell, engulfs a bacterium, okay, and then it chops it up, as you guys have seen, and basically it takes the little pieces of its fragments and it puts it on the outside, and it presents fragments of that antigen on its MHC2 protein complex, not its MHC1, because think about it, if you put that on its MHC1, you're marking it for what? Death, and these other cells, especially these natural what? Natural what? Killer cells, NKS, would just kill them. So these guys are already lurking too, right? Remember we learned about them? They're part of the innate system too. But they're looking for what? Something on the MHC1 receptor. These guys put this on something called the MHC2 receptor. They're professional antigen presenting cells, and they're there for a reason. Now, this happens, so this would not kill these because these are on different receptors. But this would kill a sick cell that's being infected by bacteria because it would present part of those antigens on the MHC1 complex. That's the complex that basically measures whether that cell should be dead or not. We do not want to kill this cell. This is important you understand this. Now, whether we're not being overrun or not, this happens. If we're overrun by these, the first wave can't handle this. what happens essentially is that this is going to work its way into something called the lymph node, the lymphatic system. Okay, now this is really important. We have, let's say, our heart muscle, and basically we push out blood through the artery to eventually capillaries, very small places where cells can get nourished. And then, of course, that gets bigger and goes back through a what? A vein back to the heart. But what people don't talk about is that there's spaces between the blood and where cells are. We have lymph that travels through them. Now, lymph is a fluid. It's not blood, but it's sometimes fluid of the blood that goes through these interstitial spaces where cells are. And this lymph will collect in a lymph node, and it connects back into the circulatory system. So the lymphatic system really is a filter for the blood. But really what it's there for is for our white blood cells to move throughout our body. So this is a little diagram of where we have some major lymph nodes in our body. Okay, now, and these little nodes here are where we have major areas where there's a proliferation or storage of the adaptive cells. Now, these are called leukocytes. Okay, these are white blood cells that love to attack. They're phagocytic. Okay, and the adaptive are called lymphatic system or lymphocytes. Now, lymphocytes mean that these guys reside in these lymph nodes. Now, understand, these lymph nodes are connected to the bloodstream, but they're able to travel throughout a lot of these what? Cells. It gives ability for the white blood cells to collect and go through parts where the bloodstream isn't. Okay? Although it's still part of it. Now, very, very important. So, we've had an infection. We have a dendritic cell, and it's just one case. There's other types of cells that can professionally present. It engulfed the antigen. Pieces of it are cut up. Those little pieces are put on the MHC2 part of the receptors so that we don't kill this cell. Because we want this cell to eventually work its way into a lymph node. In the lymph node, we have T and B cells. And these are part of the adaptive. We have T and B, and these are white blood cells. Now they're called T and B because the B cells are made in the part of our bone here, are what? A marrow of our blood, bone marrow. So these are developed in the bone marrow. Now T cells are also made in the bone marrow, but they get developed in the thymus gland. That's a little gland on top of the heart here. That thymus gland, they get pretty much finished. Their production is finished. We'll talk about that in a little bit. So these T and B cells are here, waiting. Now this is, to me, the most fascinating part of the entire thing. When I first learned about the immune system, I always thought that anything that we encounter, we develop a resistance to. So if we see this bacteria, we develop an antigen, or we develop an antibody, or some kind of resistance that fights it. But we have to see it first. I always thought that. But this is not how we work. To me, it's fascinating that what we've done is that we have built T and B cells just by random DNA splicing. Now, there's 30,000 or so genes in our body. Yet we make billions upon billions of different T and B cells with slightly different receptors. Now, the B cells receptor is different. It's actually an antibody. Whereas the T cell, okay, has a very small receptor looking for a little piece that's going to be attached to this presenting cell. So let me explain. So when we make the receptor for a T or B cell, the body just normally takes the little receptors, and you have some common areas, but then you have some constant areas and some ones they jump around. They basically splice that gene of proteins, of amino acids, randomly to make billions upon randomly different receptors, with the hope that they're making a receptor before they even see the antigen. I'll say that again. They develop these T and B cells who are made by either an antibody for a B cell or a receptor for a T cell, totally randomly. They make so many random arrangements that just by chance, something that enters our what? System will have a match for. I find it to be absolutely amazing.
But because there's so much, don't they have to try to match with it?
Right, and that's why there's not a lot of them. So that's why the first response is very slow. Maybe out of 10 billion, maybe there's 3. So what happens is, and I'm just making up a number, I don't know for sure, but this presentation or this professional antigen presenting cell that chopped up the bacterium and presented tiny little fragments for the T cell to what? Find. It makes its way through the interstitial space back into the storage area where there's T and B cells. It moves around, and just by hook or crook chance, one T cell finally matches with it. That T cell gets activated, binds to it. Now you may say, Mr. Grodsky, why does there have to be a presenting cell? Well, think about it. We are making all of these T cells that are independent upon knowing what we're going to find. If we were to cut up this antigen in tiny little pieces, isn't there a good chance our random, basically key factory will fit these things eventually if I present a lot of them and all their different foreign proteins at the cells? The answer is yes. If I kept these proteins intact, well, you can't attach to those big proteins. If I cut them up in tiny little pieces and present them differently, there's got to be something that we've made randomly that'll fit with that. That's why it's done that way. So this gets into our lymph node. It stores the T and B cells, and a T cell will link to the MHC2 receptor. Okay? It'll give off cytokines. What are they? Those are basic proteins that stimulate other B cells. Or this activated T cell randomly will bind with a B cell who's made independently, and billions of them. So the T cell, sometimes called a T helper cell, okay, will bind to this MHC2 protein complex, gets activated, produces those proteins that are released. Those proteins released will hit a B cell that's specific for that what? Receptor. Or this T cell itself will hit the B cell. Now, once the T cell is activated by binding with this receptor on the MHC2 receptor place, it proliferates. Clonal proliferation. What does it make? It makes more T cells that are specific for this antigen. Only. So what you got in the node is you've got T and B cells. Each one of them have different what? Receptors. Most of them don't even get used. But just by chance of making so many of them, one of them, ding, hits the jackpot, fits to this connector, gets excited or gets activated, and it proliferates into billions upon T cells that are unique, that are all the what? Same receptor. Not only do we make some more helper cells, we have some what? Effector cells. What are the effector cells in the T? They actually do the...
They secrete the antibodies?
No, not them, not the T cells.
Okay.
You have other T helpers, but you've got those cytotoxic T cells. Those are the ones that get produced, that get pushed around the body looking for that particular antigen. So now they are going to look for the antigen on the MHC what? 1. They have a connector looking for any cells that are under attack by the MHC1. Why MHC1? We presented this cell on the MHC2 so that T cells can find them. We proliferated them. We made more helper cells that can do what? Okay, hit other helper cells. We'll talk about that in a second. They can do more. They also make cytotoxic T cells that go around and do what? Attack any cell who's under the what? Influence of that antigen only. It kills it and moves on. How does it kill it? Yeah. You can produce granule enzymes, or enzymes that basically dissolve it, or they just puncture a hole through the cell membrane so that water and ions rush through and it just pops, right? The reason why you have that function. Okay, now, what else does it make? Memory cells. T memory cells. Now, what are those? These are T cells that have what? That have a receptor specifically for that antigen. Now, there's not a lot of them, but they are not involved in the killing. These guys don't last very long. They got to produce enzymes. They're busy cells. Busy cells wind up being what? Dead faster. Okay? So these guys don't last very long. Days. Okay, that's why the first response is going to end soon. Helper cells, well, what else can they do? Well, a helper cell can activate a what? A B cell. They can attach to a B cell. Okay? A B cell who has an antibody that matches that receptor perfectly. How? By chance. We've made enough random splices to produce enough B cells with so many different combinations that we have to have that combination as long as the what? The receptors are small enough. Okay? So, the B cell gets activated here. It proliferates. Clonal expansion. What does it make? It makes B cells that are effector cells. And these are the plasma cells. Now, the connectors on the B cells are different. They're antibodies. A little different. So what happens when B cells are activated by a T cell, they make more B cells, but they make effector B cells called the plasma cells. These plasma cells are the ones that produce the antibodies. And they produce these by the thousands. Each cell produces like 30,000 per second for like 5 days. They flood the bloodstream and the lymph, okay, with these antibodies. And when they come in contact with that perfectly shaped antigen, whether it's on the what?